First of all, this is to make sure you're in the right room. This is the monthly Exploring Ethics Forum, and we will get back to tonight's topic in a moment. I'm Mike Kallichman. I'm the co-director for the Center for Ethics in Science and Technology, and also professor from UC San Diego. The purpose of these forums, for those of you who are new here tonight, is to provide an opportunity to hear about new developments in science and technology, and then to reflect on some of the ethical challenges raised by those developments. We aim in these discussions for thoughtful approaches. We tend to try and pick topics that often are very controversial, sometimes very well-known controversies. We've talked about human embryonic stem cells, for example. We've talked about climate change. Tonight we'll be talking about a controversy that is probably not as well known to most people. One of the things that I recognize as I look around the room and see a number of people here is that we are, given tonight's topic, which has to do with HIV, we are probably challenged somewhat because I understand from some of my friends and colleagues that there was a rally scheduled tonight at 6 o'clock, regardless of the outcome of the decision earlier today, that at this point reverses the vote on Proposition 8. So based on that, a number of people who might have been here tonight are probably there instead of here. But having said that, how many of you are here for the first time tonight? So it is still about half the audience. So those of you who are new, you will notice that the other half are back here again, so there's reasons to keep coming back again. A couple of things, logistics-wise, I want to alert you to is before we finish the program tonight, we are really attentive to what our audiences are looking for in format and content. So those yellow evaluation forms would be greatly appreciated. Either hand them to Shannon or to me, or leave them on the little table by the door before you leave tonight. And that way we can get a sense of what's good, what's bad, what you'd like to see, and we'll take a close look at that for future programs. And I think those are all of the issues I want to cover definitely right now, other than to acknowledge at the beginning that this program is possible only because of the Fleet Center's generosity in allowing us to be here monthly. We've been doing this for over 2 years now, every month, and it's been an excellent partnership, which we hope to continue. So for tonight's program, the speaker you will note from your programs, which are on your chairs, is Dr. Susan Little from UC San Diego. And the way this program came about was because Susan was looking for funding for a research project. And she didn't come to me to ask for the funding, because I didn't have any, but she did come to me to ask about the ethical dimensions of the project she was working on. And I found it particularly intriguing. And I'm going to leave it to Susan to explain some of the background that goes into this, about what we know about HIV and AIDS, about what we know about the virus and how it changes, and what some of the implications are from those changes that occur. Maybe there's one last question I will ask before we go on. How many of you heard the program today on These Days in KPBS? So that's a fair number of you, and you still came tonight, so we weren't too bad. So Susan and I were on that program. So I want to turn it over now to Susan. She will make a presentation of about 25 minutes to half an hour, and then we will have the rest of the time to discuss the ethical issues here. And I want to make it as clear as possible that I think neither Susan nor I believe we have the right answer to the challenges that we're going to be raising. So we're seeking answers and contributions to this conversation. So Susan, I'll turn it over to you.
So, as Mike said, thank you very much for coming. Give me 30 seconds to turn this over. For those of you who are missing the event tonight, in my nonpartisan way, I thought I would at least say, for those of you who are on the no on Prop 8 side, it's been a good day for you. Okay, so, oh, thank you, that'll help some. So, by way of background, I thought I would start with a couple of slides to sort of set the stage with why the subsequent discussion is even important. Beginning with the number of new infections that are occurring, new HIV infections that are occurring in the United States. And though you can't see it, this begins in roughly 1980 all the way through 2006. And shown here by risk group, men who have sex with men, you'll see this abbreviation, MSM, injection drug users in pink, heterosexuals in blue, and men who have sex with men and use injection drugs in this orange color. And what you can see here is after a known very high peak of new cases among men who have sex with men, sort of numbers drop down. But what is quite concerning is that since roughly 1990, all of the risk groups, the numbers of new infections have remained flat or actually declined, other than among men who have sex with men, where since 1990, the number of new infections has gradually increased. So despite all of our prevention dollars, treatment, access to care in this very developed world, we still have increasing numbers of new infections in this risk group. Next, transmitted drug resistance in the United States is a significant concern. Starting down here, transmitted drug resistance is, if there's too much feedback, let me know, it sounds loud up here. Transmitted drug resistance is the infection of a newly infected individual with a virus that is already resistant to one or more antiretroviral drugs. That individual, therefore, has fewer treatment options when they need to start treatment for their HIV infection. And if you look at all newly infected or newly diagnosed people in this study, and here they looked at roughly 2,000 individuals out of 10,000 newly diagnosed people with HIV across 11 surveillance sites in the United States, about 15% had significant transmitted drug resistance. And if you look by men and women, about, again, the largest category of newly infected or newly diagnosed individuals, men who have sex with men, again, about 14% of men who have sex with men had transmitted drug resistance, and about 14.5% of heterosexual women, the two largest groups by number of individuals in each sex category. So transmitted drug resistance is a major concern in the sense that once someone has transmitted drug resistance, they are newly infected with drug resistance, that is the only virus present in their body. They then have only one virus to transmit on to others if they transmit virus. So as I go forward, I'm going to talk a lot about genetic analysis of HIV. So we're going to look at the genetics of HIV and compare genetic evolution. So what are the genes we're talking about? We're talking about HIV drug resistance genes. It is now standard of care in the United States that all newly diagnosed, newly infected, newly diagnosed, and people in care for HIV treatment on medications get drug resistance tests. They are standard of care, routine, regularly performed. And when those tests are performed, this is a drug resistance test, we, medical providers, get access to a sequence of the HIV gene. And that is very commonly, almost universally, the segment of the HIV gene that we're talking about when we talk about doing genetic analyses. It doesn't have to be, but it's overwhelmingly the most common because it is part of routine medical care. So tonight what we're going to talk about are these topics: HIV transmission and evolution, phylogenetic analysis, which I'll define. How do we prove HIV transmission? Can we prove HIV transmission? And in the particular context of an HIV network, personal privacy, and prevention opportunities. So I'm going to start with a hypothetical town. I called our town Impulse. Because that's how it all begins. So person A has sex with person B. Boy meets girl. Then person B has sex with C. Now C has sex with D and E. So this is a typical sexual network. Unfortunately, we also have this as a typical sexual network. C can have sex with D, E, F, and G. G, and H. So this is an equally typical sexual network. So when we talk about sexual networks, this is what we're talking about. The way that we know that this is a sexual network is because they tell us. So we don't have cameras. A and B and C and D and all of these individuals, by virtue of survey or questionnaire information, will tell us who had sex with whom, and we can reconstruct this image. So now we're going to talk about an HIV transmission network. If A is HIV infected, now A transmits in HIV infection to B, and then B to C, and now what if C transmits to D, G, and H, but not to E and F? So this is an HIV transmission network. This is what we're going to talk about today. So one of the first questions is, at what level of transmission do you, each of you, become concerned and feel like maybe the public health department, primary care providers, or the general public should be aware that a lot of transmission is going on? 1 transmission, 3 transmissions, a whole lot of transmissions? At what point does this become something that perhaps a higher level of involvement might be appropriate in order to try and intervene and reduce transmission? That's part of our discussion. This is what we might classify as a serious HIV transmission network, in the sense that a whole lot of people are being infected. And I've drawn arrows, but for the sake of argument, we often don't know which way transmission has occurred. So the questions here: what if C transmitted HIV infection to 10 people? Is that less alarming than, for instance, if C transmits to 50 people? Does that become a crisis? What if C is also infected with very drug-resistant virus? Is that what tips the scale, and now it becomes alarming? These newly infected people, as I just said, will have very often reduced treatment options. Can we be certain, however, that C transmitted to D, G, and H? I drew arrows to imply that, yes, that was true, but I'll show you in a minute, it's not always straightforward. How do we know that D, G, and H weren't infected by someone else or each other? What are the legal implications of proving or knowing that transmission occurred? How can we be certain that C infected D and D didn't infect C? Do these studies risk incriminating individuals by linking HIV transmission? Who is supposed to help limit transmission, HIV transmission? The public health departments, primary care providers, individuals? What can be done to help? Can we provide education, antiretroviral treatment, target these networks with specific, what I would consider appropriate prevention interventions for that specific programs, abstinence, condoms, a whole variety of prevention interventions are available? And then finally, how can personal privacy be maintained and specific interventions still be delivered should we decide they're appropriate for specific HIV transmission network? So these are a whole list of questions that I'd like you to think about as I move forward with the next few slides. So again, a bit of background. HIV evolves quickly within every infected person. This can be measured by looking at the genetic code or the sequence of HIV. And as I've already mentioned, for the most part, we're going to be looking at the sequence that is the gene sequence that is used to measure HIV drug resistance, because it's already available as part of routine medical care. HIV evolves so quickly that each infected person has a nearly unique HIV sequence. So sequences sampled from different people within a community or within whole cities or nationally that are unusually genetically close could signify a potential transmission event. That is, that one person transmitted to the other person, if their sequences are really too close, that could be explained by random chance. Or that they belong to the same risk network, that they may have both been infected by the same person, for instance. So phylogenetic analysis. Phylogenetics is the field of biology that studies and identifies the evolutionary relationship among the many different kinds of life on Earth. Phylogenetic analysis calculates the differences in genes to estimate their relatedness. How alike or different are they? It cannot create a definitive match or proof. So that's one of the very key things I want people to remember. Right now, today, the best ability that we have looking at likeness of genes, we can't prove based on phylogeny alone that one person definitively transmitted to another. It can't prove direction, it can only prove likeness. So although HIV can be nearly unique to an individual, it is not like human DNA or fingerprints. People have seen a lot of television and are used to thinking about human DNA in courtroom situations. HIV DNA is not the same. It is nearly unique, as opposed to human DNA, which is completely unique to each individual. So phylogenetic trees are illustrations that represent the results of the phylogenetic analysis that we just did. And these show pictorially the genetic relatedness. So this is a sort of randomly pulled family tree off the web of two people who I have no idea who they are. But it's a two-generational family tree meant to illustrate the point that here, mom and dad, William and Rebecca, had 4 children. And then these two actually had 3 children, sorry. And then George marries Cassandra, and they have 8 children. And the point here is that if you look at a phylogenetic tree, just like this family tree, you can infer that George and Edward are genetically more related than George is to Eliza, his cousin. So the people are more related to their ancestors back toward the root of the tree, who are back here, than they are moving laterally across back here. So the most common ancestors are the most genetically related, okay? So that's on a small scale, a 2-generation family tree. This can be taken to much more complex, large scales, as was done here in Montreal. In Montreal, Luma Brenner looked at all of the primary HIV-infected individuals in the city of Montreal. So these are hundreds of newly infected individuals, and found that there were the majority of individuals who did not cluster. And this is shown in a circular tree. It's just a different representation of a tree so that you can display hundreds of sequences on a relatively smaller scale. But the long branch lengths show a longer period of evolution and the absence of genetic relatedness between these sequences. As compared to over here, these are the clustered primary HIV sequences. And what you can see is that there are clusters of, I can't count these, but 10 to, 5, 10, many more, 20, clusters of sequences with down here at the terminal ends, very short branch lengths. And those indicate genetic relatedness. So what they've done is they've looked at the entire city of all of the sequences that have been contributed, again, based on resistance testing, to look at clustered versus not clustered, and then they later went down to break these out into geography across the city. So what are the limitations of phylogenetic analysis? I've already mentioned that current techniques are not reliable enough to estimate the direction of transmission with certainty, and that similar strains may be found in many more than 2 individuals, especially if they're part of the same transmission network. So just as Dr. Brenner's study just showed, you can see a cluster. There's no limit on the number of individuals in a cluster. Just because one individual transmits to another, that doesn't necessarily define a cluster just because 2 people transmit. You can have a cluster of 75 individuals. It just depends on the activity within the network, sexual or drug use, whatever the mode of transmission is, and the efficiency with which that virus is being propagated within the network. So we're going to return to our transmission network. So I last showed you this transmission network, which looked very simply that transmission occurred here. It looked like B was infected by A, then C was infected by B, then C infected D, G, H, but not E or F. So that's what things look like on the face value. And again, when I say face value, I mean B comes in and says, I slept with A. A is infected, so we often infer that transmission occurred in this direction, particularly if we identify this person after we identify this person. But in reality, you can then do sequence evaluation and things look very different. So we can find out, for instance, that A and B are infected by the same virus. But as I just said, we don't know who became infected first. We may know that this person presented first, but that doesn't tell us anything about who was infected first if we don't have a date of infection. C, we know now, was infected by a third party, or I should say, has the same virus as a third party, but that's genetically unrelated to this individual. So although they had sex, no transmission occurred. They have completely distinct viruses. This person shares a virus with this person. So C then probably goes on to transmit or share the same virus with this person. And again, no transmission here. And then H is already infected by a strain of virus that's the same as is seen over here, but has no known association to these two individuals. Same network, but now alternatively, A could have transmitted to an unknown person who we have not yet sampled. So B is infected, A is infected, and actually transmission went through this third person and never went directly between these two. So they could still have the same virus and look for all the world like transmission was direct, but in fact it went through an intermediary or even two intermediaries before it came here. And then C is infected still by this person, H. H could have gone on to infect G. G could have then transmitted also to an unknown person that we haven't sampled, so we don't know who this person is in our network who infects D. So C is sitting here in the middle of this very high-risk transmission network, and despite being in the center of the network, has actually transmitted, as best we understand, to no one. So this is part of the challenge, is that it can be a big challenge to look at these networks and say, C is doing very bad things because he, in this case, he doesn't have a red dress on, is transmitting to all these people, when in fact, you always have to remember the unsampled, the missing individuals. There are people out here who may be transmitting to people in our network who we have not sampled. So until, if we ever do have the ability to know the direction of transmission, all we're doing is trying to say we have a high-risk network, we know a lot of people in this network are transmitting. Would this network benefit from some targeted prevention, intervention, and or testing so that everyone in the network knows their status? So, for instance, again, just to make things a little easier to comprehend, let's go back to the basic transmission network. What if A, B, C, D, E, F, G, and H, all of them, are associated with the same club or chat room? So now they're a venue-specific transmission network. Would there be an opportunity to intervene with a specific targeted message that might reduce the risk that these two individuals become infected and reduce the risk that these others transmit infection and potentially get some of these individuals into care who are infected and don't know it? I haven't mentioned already, 20% of the people in this country who are HIV infected don't know their status. So they're HIV infected and unaware of their status. So simply testing people is a huge prevention intervention. So I've already alluded to this. Could a prevention intervention help to better identify potential known sources of transmission, A and B, unknown sources, these question mark people, of HIV transmission, and people at risk for acquiring HIV infection, ENF. There are public health examples of these sorts of public health tracing programs. The public health prioritizes treatment and tracing of contacts for people with both active tuberculosis and syphilis. And there are policies and procedures that are already well established and in place for protecting patient confidentiality when these sorts of programs are put into place. There are furthermore state laws that generally protect the confidentiality of all personal health information, sexually transmitted disease information, including HIV and AIDS. However, one of the challenges here is to balance individual privacy protections with legitimate communal uses of health data. That is what we're talking about here today. You know, there's this concern about the absolute imperative to protect personal health information, but what if we had the potential benefit to stop or interrupt a serious HIV transmission outbreak by utilizing some personal health information within the public health sector? At what point can we sort of tip the balance? What access to what personal health information would be acceptable in that balance? So confidentiality is also subject to practical limits, including the possibility that partners might guess the identity of the index patient at any point during the process. So just to illustrate, person A here says, I have a sexual partner. I want the public health department to notify my partner anonymously and confidentially. So the health department goes and notifies anonymously, I shouldn't say anonymously and confidentially, anonymously notifies this partner B. Well, if A only has one partner, B may very well guess that it was A who told the public health department about their relationship, and guess, in fact, that it was A who came down with the sexually transmitted infection that they have been exposed to. So prevention measures cannot be a one-size-fits-all. We need to use network analytical methods to characterize HIV transmission dynamics within a population. That's really our goal at this stage, is to look at how we can use these techniques on a population basis. We want to use network and phylogenetic methods to identify risk groups, venues, and locations, not individuals. We want to focus our outreach and prevention programs on well-identified hotspots, whatever we identify where a substantial amount of risk seems to be occurring, for instance, bars, clubs, chat rooms. And since network dynamics change, our network surveillance needs to be dynamic. And then legally, the identification of individuals who have transmitted HIV can have significant legal implications. Therefore, the use of these data for any public health purpose must include decriminalization of unintended HIV transmission during consensual exposure, and a legal recognition that phylogenetic linkage using HIV sequences does not prove beyond a reasonable doubt that transmission between partners occurred, such as with a third-party intermediary. So just because person A and person B have the identical sequence does not mean that transmission could not have occurred with a third-party intermediary who actually transmitted virus between these two. So in terms of prevention opportunities, we would like to encourage HIV testing within the risk network. I've already said 20% of people don't know their status. That's a huge prevention opportunity right there. So if we could target our testing resources in high-risk networks, that would be a huge advantage. We need to identify HIV transmission risk networks and provide targeted interventions and prevention education. We don't have endless prevention dollars. So if we had some prevention dollars and we could spend them more efficiently in networks where we knew HIV was being transmitted and we knew there were individuals who were at high risk of becoming HIV infected and at high risk of transmitting HIV infection, that's where I would rather spend my prevention dollars. And the prevention intervention itself would certainly vary depending on the risk network. It might be needle exchange, antiretroviral therapy, risk reduction counseling, condoms. It would vary potentially. And then finally, individuals with drug-resistant virus can be counseled about the risk of onward transmission. New infections within that network could immediately be counseled about appropriate therapy as opposed to being started on inappropriate therapy, waiting for them to fail, and then finding out, oh yeah, you were infected with drug-resistant virus from the start. And then finally, we'd like to maximize treatment options for all newly identified individuals with HIV infection and transition people into care as soon as it is possible. I should mention the new HIV treatment guidelines were just released last week, and although there are treatment indicators, that is CD4 and other clinical indicators for therapy, it is now recommended that antiretroviral therapy be considered regardless of CD4 cell count. So there are no contraindications across any CD4 cell count at which we should not at least be considering antiretroviral therapy.